So, Scott, what's the latest in your research?
Well, so I think that's a good. This is a good time for me to do an intro. That's a good idea. While Michael's getting his audio working. Yeah. So if I can give a background so that everybody knows where I'm coming from and what my interests are. My background's primarily in genetics, although I've been a part of a bunch of different research areas, and I developed MECFS myself back in 2017. It was literally a week before I was going to grad school, and I was handling a chemical in the lab, and following this chemical exposure, I developed MECFS, and for a while I was in pretty rough shape, and I started to get better and improve, and I got back to work, and I did some consulting in MECFS space for a few years, and I also was a scientist at a genomics company for a few years as well. And following a move to a new apartment, I learned that I had multiple chemical sensitivity as well. And, dealing with some mold and paint and things like that caused my health issue to get a lot worse. And so I dropped out. I had to leave my work there. And I started doing kind of my own just personal research, for a couple of years, and I was working on imaging and specifically electron microscopy. That was a big focus of mine. And I had some really interesting results. And I also made some really interesting connections through my personal experiences. And then I spent a lot of time speaking with other members of the community and found that they were having similar experiences to what I was having. So for all of essentially 2024, I've been working on advocacy and spreading awareness for specifically environmental factors driving MECFS, which I almost never hear people talk about. I hear some people talk about mold, but not other factors. So it's something that I think is under researched, underfunded, under considered. I say under. I think essentially nothing's happening at all, almost. So it's just an area that I feel should be picked up, because if we're going to have a really good roundabout approach, we've got to look at a lot of different areas. Um, and I do have a paper that I co-authored on multiple chemical sensitivity that was submitted about a week ago to be published. So I'm excited about that. I did a podcast interview about a week or so ago that hasn't been put out yet, and so I'm looking forward to that coming out. And this is the first ever panel discussion I've had, and I've had it because I've joined a bunch of spaces on X this year, and I always get a lot of people asking me questions about science and research and what does this mean, what does that mean. And they're really good questions, and a lot of people are interested. But sometimes it can be disruptive when somebody's trying to hold a space about something else, and I draw attention away from the chat. So I figured it's a good idea that I hold my own space. And I held one about a month or two ago, and I had a lot of people show up for that. And I'm like, well, this is going well. I should invite some other people in that can share different ideas and perspectives. And I thought this is a really good opportunity because I don't think the patient community gets to hear from researchers enough. When they do, research presentations that they have are very tailored and focused on like one specific area. And because of institutional affiliations, people also like they don't have the chance to kind of speak freely and share speculative thoughts. Like I think maybe this could be going on. We should pursue that in the future. They really only talk about like concrete things. And I think there's a time and place to that we should have to be able to talk about theories and speculative ideas, because we're like when we're dedicating research funding to certain areas, we're using resources, we're burning through resources, so it makes the most sense to really have intelligent, targeted research studies be done, and really open conversations are required for that. And also, it gives us a chance here to, answer questions from the community and speak in terms that, just average people can understand that don't have a technical science background. So I really wanted to have like an open discussion today as much as possible, and just get people to share their ideas and thoughts and opinions. But I do have a bunch of questions here that I have from the community that have generously submitted a bunch of questions that they would like answered. And I came up with some of my own questions as well, targeted specifically to each of you because you have very really good but specific experience and backgrounds. And I think having a bunch of panelists on it is the best opportunity to hear a bunch of different research ideas as opposed to getting hyper focused on one thing too much. We have a bunch of different backgrounds. So I'm really excited for today, and I appreciate everyone taking the time to come out and listen. And I hope it's really informative and helpful for everybody. I just want to check on Dr. Scoma, how he's doing before we get started. Yeah, he may be having issues, so I'll just get started here and hope he can join in later. If you just give me one more minute here.
Oh, what was the chemical, Scott, that triggered your multiple chemical sensitivity?
Yeah, the chemical I was working with is called hydrazine. And interestingly enough, if you know, I had a conversation a while back with Eric Johnson, who's a big mold avoidance guy. You probably seen him here on Twitter. He's really intelligent and he's learned a lot of things over many years. But in talking with him, he actually had an exposure to hydrazine himself. He worked on a military base in Germany, and hydrazine's used as rocket fuel, and so that was one of the things that he was exposed to right before he developed ME/CFS. So yeah, it's really interesting. There's not a lot of reports on that compound specifically because it's really only used as rocket fuel. I happen to be using it in the lab in a specific experiment, but it's a pretty limited thing. Yeah. But okay, so we'll get started here. And I wanted to just give an intro to each of the panelists here, let them kind of talk about what their experience is and hear from them. And I've got a few questions for them specifically too. So kind of have like a little mini interview to get started here. So Geoff, I guess I could start with you. And you are a scientist who is focused in toxicology. You have a good background in chemistry as well. And I see you do a lot of advocacy on awareness of endotoxins, and that's something you're really interested in. So if I could just get a little bit of a background summary for you, what your research experience is, I'm sure everybody would love to hear.
Yes, thanks, Scott. I studied biochemistry at university over 50 years ago, so things have moved along since we were chopping the heads off rats and doing enzyme analyses and so on. And later on in my career, I worked in a genomics laboratory and became very interested in looking at disease relationships to DNA. But the laboratory that I worked in was mainly focused on inherited diseases, and I was much more interested in environmental exposures as triggers of disease. And so I helped to balance out the research group that way. And at one stage, I was flown to Canberra to participate in a national conference in multiple chemical sensitivity, which I prefer to call chemical hypersensitivity because each individual responds differently to levels, and there are orders of magnitude of differences in sensitivity to the same chemical exposure. So I became heavily involved in analysing the Pfizer documents that were released by court order in America, which were meant to be buried for 75 years. So I was in the research group, the collaborative group, who got the first copies of those documents. Some of them had to be converted from scans into searchable OCR PDFs, and that helped greatly. And initially I was interested in the symptoms of COVID, and then when I learned from the Pfizer documents what was in the jabs, I was initially concerned about some of the so-called novel chemicals that were being used, but one particular document aroused my interest, and it switched my focus to endotoxin as the universal contaminant of all vaccines, and causing the symptoms match with all of the adverse reactions that come through. So I'm sort of straying around here, but the links between long COVID, as you say, and the CFS are very, very strong indeed, especially when we've got speakers today who have actually been analysing people's blood. And so I'll be looking forward to hearing their results. But the sort of things that I've been reading about are CD14 and that sort of thing. So anyway, it's very, very, the need for multidisciplinary teams, as you say, is absolutely vital.
Yeah, well, I kind of wanted to ask you a question about that. So your knowledge of vaccines, specifically in that area, I know there's a lot of vaccine injured people since the pandemic started, and there's a tremendous amount of awareness for long COVID, but not much for these vaccine injured people, and they're really kind of confused and wondering what caused this, and a lot of people think an immune insult is likely responsible. So I'm just curious what your take is, if you have any ideas on why people have developed chronic illness from these vaccines. Like, do you have any idea as the mechanism of that?
Yeah, my primary focus is the fact that when you are jabbed in the arm, and it doesn't have to be a COVID jab, any jab will do, that will actually trigger and exacerbate gut dysbiosis, and that basically involves flooding your blood and lymphatic systems with remnants of dead bacteria entering through your damaged gut lining. And we know that chronic fatigue syndrome pre-COVID, of course, was directly linked to viral infections. That's a very, very strong connection. And viruses are known to disrupt the gut lining, and some of them reside and breed in the gut, and then they move and they actually stay in the body. There's a very strong link between the herpes virus and chronic fatigue syndrome, and that can be reactivated. So it became obvious very early on that when you were jabbed with a COVID jab, herpes was one of the very first viruses to be reactivated, along with Epstein-Barr and various other dormant viruses within the body. So the trigger for that is always endotoxin, and that's easily demonstrable both in vitro and in vivo. And so I'm interested in triggers. The thing about the gut dysbiosis is once it's established, it's very difficult to calm it down. So you then move from the instantaneous effects to the chronic problem. And you can actually, we were chatting in another space that if you suspect that you've got gut dysbiosis, you can actually perform a relatively safe test of swallowing two different sugars, one of which is metabolized and one of which is eliminated without touch through urine, and the levels of those sugars in your urine can give you an index of how leaky your gut is. And so people who picked up CFS well before the COVID era and may have had it exacerbated by the COVID jabs should look primarily to the gut axis, in my opinion.
Yeah, I have personally even heard about people developing their own personal vaccines using antigens from bacteria that they collected from their urine, and there's a whole protocol for doing that. I'm not an expert in that area, but obviously, ever since like a decade or so ago, when the gut biome became a big thing, that's been a big area of focus. And I've heard a number of patients have tremendous improvement from fecal matter transplants as well. So that's certainly an aspect that deserves good consideration. I just wanted to just keep moving so that we get all our panelists introduced here. Dr. Scoma, I see you're on now. Just want to check with you. Is your audio working?
Yes. Can you hear me okay, Scott?
Yeah, absolutely.
Excellent. Good.
All right. So Dr. Scoma he's a private practice infectious disease clinical immunology physician. So he's got a lot of experience that long COVID patients are particularly interested in. But he has personalized care for ME/CFS and long COVID. And that's really important because I hear that he spends a lot of time talking to individual patients at all sorts of hours of the day. And he gets a good idea of what people experience, not just during those one-on-one doctor's visits, but around the clock, what these patients are going through. So Dr. Scoma, if you'd like to give an introduction about yourself and talk about your practice and experience with ME/CFS and long COVID.
Thank you. Sure. Yes. So as you said, I'm private practice infectious disease immunology for the last 17 years. A vibrant practice within the inpatient realm as well as the outpatient realm. And in much more recent years, gained such an appreciation as to the complexity that which is ME/CFS, the multiple the multiple etiologies likely encompassing it, from the vasculitic, the neuroinflammatory, the immunomodulatory. And then seeing that uptick, not only then that, but then, dealing with the initial COVID cases in the hospital setting in 2020, and seeing the, just the destruction that, this virus caused upon not necessarily even immunocompromised hosts, but all hosts. And then now seeing the sequelae of, a post-sequelae syndrome. And just in terms of just being able to try to being up to date with the research, trying to understand as best as possible the pathophysiology and the mechanisms behind it. Because if you understand that, then it's a better chance of at least what we best have now, or, mitigation of symptoms, mitigation of disease processes. And that said, with that, in taking care of this patient populace and the things that go with it, like mast cell activation syndrome, dysautonomia, oftentimes some of its relationship to chronic tick-borne infections and co-infections. It's an ever-changing kind of paradigm where, to say that you need to know a patient, quite intimately from the very, very beginning, how things started, what got them sick, what was their medical history like, what's their current history like, their social constructs, what have they been on, what has worked, what hasn't worked. It's extremely interesting, and it's an extreme passion of mine, particularly when you are able to mitigate these symptoms and mitigate the disease process, obviously depending on the patient, how sick they are, how severe they are, how long they've been sick, how many issues are underlying. It's extremely gratifying, and it really essentially moves me more so than anything else.
Well, I wanted to ask you about the kind of treatment that you're able to provide, because there's obviously not a lot, or virtually no approved treatment for ME/CFS, for example.
Correct.
I mean, there are some recommendations from some organizations in terms of managing pain with low dose naltrexone, treating POTS with electrolytes and stockings and things like that, which is a common comorbidity. But what kind of things have you seen in your practice that has been beneficial to patients that maybe potentially differs from what larger organizations are just kind of broadly recommending?
Right. So when with respect to low dose naltrexone, low dose Abilify, some of the sleep aids when you're dealing with the insomnia issues or the fragmented sleep, like low dose doxepin, old school first generation antihistamines such as even Benadryl, Unisom, hydroxyzine. It's really kind of in terms of how I approach it. It's really with respect to what are the patient's symptoms? First off, how long has the patient been sick for? What's the severity of illness? What's the degree of illness? Is this somebody who's mildly functionally impaired, or is this somebody who's bedbound for 3 years, 26 years old, say, with severe ME/CFS, whose feet have not touched the ground in 3 years? Very, very different patients, different outlooks, and so on and so forth. So
So
generally
you've
speaking,
seen different treatments be recommended for different subsets?
I'm basically going off of what I think is going to be most required with respect to disease severity, disease process, and what's going on. Obviously, if somebody's main symptom is, severe dysautonomia and autonomic dysfunction and, neurological issues, for example, you may utilize something, and this is just my own personal opinions. This does not constitute, obviously, recommendations. However, there's been a lot of success with peripherally acting beta blockers, centrally acting alpha blockers, such as guanfacine, pairing it up with, for example, NAC. Low dose, I kind of use like the rule of 50%. 50% of these things work well. The other 50% in other patients don't work as well. So that's why it's very much, to a degree, a bit of trial and error, because as you said, there is no magic bullet. Oftentimes, there is no true FDA approval. If I think that there's an element, like I said, of dysautonomia, for example, like I said, the alpha blockers, the centrally acting beta blockers, fludrocortisone, midodrine. If you're concerned about viral reactivation, particularly entities such as EBV or HHV6, HHV7, things that are quite overlooked. Again, many people do respond to reasonable doses of valganciclovir. I've had people where I've had to implement intravenous agents such as cidofovir and foscarnet when I really thought that was driving things. If you have somebody where you feel that the major driver is mast cell activation, then you're looking at a completely different paradigm of H1 and H2 blockers, mast cell stabilizing agents such as cromolyn, ketotifen, quercetin. So again, it's very varied, and there's really no one sort of cookie cutter approach that works for everyone. I do have generally my kind of mantras that I'm not a huge stimulant person for people with ME/CFS because it is just going to cause, generally, false energy. Do I have those couple of patients that have been sick for many, many years and they do find that, say, something like Vyvanse a couple of times a day gets them over a hump? They're not addicted to it. It's very low dose. Sure, I'm not going to, what's working, I'm not going to interrupt that. But the idea is, perpetuating a regimen that's going to give basically better functionality, better quality of life, and better symptom mitigation as evidence-based as possible, despite the fact that I don't think we have any real, great, randomized, double-blind, placebo-controlled trials. This is just what's coming out and staying, up to date with it as possible.
Absolutely. Thank you. Thank you very much, Dr. Scoma. And I just wanted to remind everybody listening, I highly recommend you drop our panelists a follow. I've gotten slowly to know everybody, and I think we have a great team today. I've heard from a couple of patients in the community that Dr. Scoma has excellent personalized care. Certainly we all know those doctors that will just dump you off and send you on your way, and Dr. Scoma absolutely doesn't do that. He's a great man, so highly recommend giving him a follow as well. So thank you very much.
Thank you.
Next, I wanted to introduce Sam Keating. Sam, are you there?
Yes, I am. Can you hear me all right?
Yes. Hi, Sam. So Sam's a friend of mine for a few years now. We've met online through an advocacy group, and I think he's a wonderful guy. He's always been incredibly polite and kind, and we've had a few good conversations. He's working on his PhD now, and as a systems biologist and bioinformatician, and he's been involved in MECFS research and a number of advocacy groups for a number of years now. Sam, would you like to give a little background on things that you've been researching and what you've been working on?
Sure, and thanks again for having me today. I was writing a few notes down just before we started here, and one thing I'll say just up front is I prefer to sort of collect disciplines and skill sets. So I've actually worked across a bunch of technical disciplines, and I consider myself a systems biologist at heart, where systems biology is really the bread and butter of specializing on interactions and connectivity in biology instead of just one domain of expertise like just neuroscience or just immunology. So I've got experience across human herpes virus systems biology specialized research from a specialized lab, phage therapy manufacturing startup that I worked at for a few years as a computational biologist, uh, and the microbiota gut-brain axis research I did for my first master's in bioinformatics. And across my several degrees, it's been bioinformatics, chemical engineering, systems biology, genetic engineering, and beyond. So I'm really serious when I say that I prefer to collect disciplines. But aiding this vision and mission is more than half my life ago, I decided to go into research and become a patient-turned researcher after I'd been referred to 50 different specialists in 2 short years, exhausted all treatment options, and had no symptom relief. And I just wouldn't take no for an answer after I was told that there were no doctors left to refer me to, no treatments left to trial, just goodbye, good luck, and you may never find relief, but goodbye, you have to leave now. Please don't call me again, I won't be helpful. Goodbye. And I wouldn't take that as the final end all. So I've been on the long path ever since to develop new treatment options someday for at least one condition that I have personal experience living with. And along the way, my focus sort of broadened from what I thought was only one rare disease that I was on my radar that I probably like, in many ways even had, since I hadn't had onsets for the more complex reality that just expanded over the years. But 2016, I ended up getting diagnosed with 6 different syndromes in a single year with my sudden POTS onset. And I just wanted to cite one figure that I see a lot of papers coming out around the 75% mark. About 3/4 of MECFS cases are post-infectious in onset. My case, however, was not. Scott, you commented that yours obviously was not. Whether you chalk it up as the primary condition being MECFS or MCS or different ways of framing it, one thing I think a lot about is how we frame knowledge and primary labels, and how we can sort of go about consolidating different knowledge streams. So I broadened my focus to comorbidities, and I've really been focusing on how would a scientific researcher running a lab, which I hope to do someday, how would one actually rigorously analyze comorbidities, since they're far too often just simplified away and just treated as confounding variables and just considered too complex to study, too complex to understand. And I won't take that for a final answer either. So I intend to continue to stake out the way to specialize in comorbidities and mapping out the interactions and connections across not only genes and proteins, but entire conditions and heterogeneous syndromes as well.
Well,
So right now, my active research is on MECFS, and I just wanted to summarize and say text mining is one of the powerful tools that I've learned along the way from my computational research skills that I'm hoping to consolidate knowledge aggregation across all MECFS biomarkers, subtypes, and statistically significant findings that have been proposed on the individual study level and consolidate into a human readable table of just here's everything that's been proposed across different research from different angles with different hypotheses and ways of framing knowledge and ways of understanding the world. How do they work together synergistically?
Well, speaking of talking about complex biologies, I just got a question for you here because I've seen briefly. I wish I need to sit down and go through it more, but you've had a couple of publications that you've put out on genetic analyses, and there's been a large number of genetic studies in ME/CFS that have been going on for over a decade now, and they all come up with these certain markers that have slight increases in predisposition to developing ME/CFS. And there's a new genetic study that I'll post in the comment section as well that shows it. I think this one was in long COVID, but it shows gene loci in immune and thromboinflammatory predispositions. And so my questions for you is, how do people like take all these different loci with slight increases in predisposition, and how do we make sense of that in terms of putting together a mechanism and finding out what might be coincidentally occurring, or what's relevant to the actual mechanism of the disease? I'm wondering if you have any thoughts on that.
Yeah, certainly. It's been very interesting looking at the genetic sort of landscape of ME/CFS research and being aware of the genomic landscape across research of other conditions. And one sort of comparison point I can bring up is that some conditions have put in enough money and genome sequencing effort to sort of reach what you can consider a saturation point, where you would actually have enough data to prove or evaluate whether there is a single gene or a single mutation that can explain a single condition. And there's, of course, going to be varied saturation points for separate conditions that have different levels of complexity and heterogeneity and beyond. But ME/CFS really has not reached that saturation point yet. There really hasn't been enough genomic research to really find out what can it look like? What is the landscape? What do we know about the genetics? What do the mutations actually look like on the population level when you can have enough data to factor in the heterogeneous nature of how complex and varied the ME/CFS experience is? So there are some recent interesting studies, and I just had a journal club with the Spark for ME Young Investigators Journal Club, where we just discussed the Decode ME paper that had used, or sorry, not the Decode ME efforts. There is an ME/CFS biobank, and then there's parallel efforts that have used the UK biobank of genetic data. And this is one on UK biobank-centric data. It's currently a preprint. And we had a good discussion this past week on it, where I think the title, I'll try to pull it up soon, something along the lines of deconditioning alone cannot explain ME/CFS symptoms and complexity. And that was sort of the central focus of their analyses. But even that draws the point that even with the same data, you could have very different ways of drawing rigorous analyses. So to fully analyze something and have it become saturated, where the process is fully examined, and we know for certain that we can form a consensus around this knowledge and answer this question with consensus confidence, we just aren't there yet for ME/CFS or long COVID. And it's going to take a lot more time, a lot more effort and money. But for what we do know about genetics and ME/CFS right now, I would say it doesn't fit the hallmarks of a genetic disorder where it's necessarily a single SNP driving the whole condition. But that doesn't mean that genetics are irrelevant. I guess I'll sort of note to my soapbox, alluding to my obsession with reductionism and countering reductionism, moving towards just embracing more synergies. The notion that a question can only allow for one answer is inherently reductive, but it's a very pervasive force that all professionals interact with. All professionals tend to interact with structures formed by reductionism. So don't think that just because genetics are in question, that if you're posing a question around genetics in ME/CFS, that it has to be all or nothing, that it has to be this is the causative factor, or is it even correlated, or is it nothing? There's so much nuance in between.
So
So
if
something can be a factor without being the driving central thing.
So if I can take from based upon the answer that you gave, you would say that probably specifically in the field of ME/CFS and long COVID, that the role of genetic studies, because obviously I come from a genetics background as well, and I understand there's a ton of bases. It's a really complicated area. There's, like you said, a lot of ways to interpret that. There's epigenetics. There's so many different factors, but the information that we get from the genetic studies might be best utilized if we pair that with other research that's going on. So, for example, if somebody's looking at clotting mechanisms, genetic research can pinpoint, certain predispositions in, certain genes that can, help researchers fine tune like exactly what step in the clotting process to focus in on. Would you say that would be a good utilization of genetic studies?
My connection broke up a little bit in your last sentence. Could you repeat just the last sentence or two?
Yeah, I was just saying, do you think that would be the best utilization of genetic studies to pair those the information that we get from them with research that other researchers are doing? So if somebody's focusing on clotting specifically, have the genetic studies pinpoint, okay, there's a mutation in this gene that identifies that this particular step in the clotting process could be a problem, that you pair the genetic studies with other research.
Yeah, and I think that's something that can really touch on sort of this interesting paradigm that we've reached that's sort of shifted in recent decades within scientific research, of sort of the question of whether you would prefer to approach a study with sort of a targeted approach versus a broad strokes, just measure everything, not necessarily need a hypothesis that we think it's this driving that, but instead to just go in with multi-omics or one of the omics technologies. So like genomics or the genome to break down what ohm means in general. Genetics and genes are building blocks of genomics. And a genome is the full collection of one's genes and DNA. Genomics is sort of a high throughput process that can measure all DNA or all genes in a single go through. So a single read through you can get for around the level of complexity and cost that it used to take scientifically to measure one gene at a time. We can now measure a whole genome. And in parallel, similarly, for proteomics, metabolomics, transcriptomics, there's multiple of these omics where we can actually measure and do research on all proteins present in the sample that we can pick up with the best technology currently available, which is a moving line over the years. Um, but to actually be able to do everything as an omics approach has really caused this different paradigm to arise, uh, where it really has just taken the ME/CFS research by storm. If you study the literature and take a look at just how many studies, just how many of the papers actually took an omics approach. Um, I am a systems biologist, so it's sort of the bread and butter of systems biology to do high throughput biology with bioinformatics mixed with the systems perspectives, uh, of actually looking at everything and not just one thing at a time or just one hypothesis, period. Um, I think it's really interesting that it sort of looks like there's some fields that have benefited from an omics sprinkled in approach where some studies have focused on omics, whereas I think MECFS might have gotten slowed down as a field as far as research, where there's so much that was poured into the omics without any hypothesis, without any targeted approach of we're going to measure all the proteins, but we think this protein might be driving this mechanism, or we're going to validate this metabolite that has been proposed to do something functional in MECFS, or even some other condition that might overlap with symptomatology. And there's just not enough horizontal validation going from no knowledge to something that can actually validate in a different combination of factors. whether that's a different condition than the original claim or a different cohort that's larger. There's so many different ways you can recombine the combinatorial process. And I think that we need to look more at, exactly as you phrased in the question, combining data types and data approaches across different types of expertise, different professionals, different data types of methodologies. I think we need to combine a lot more of it. And I think moving towards open source and decentralized science is part of how we can get closer. And of course, I am computationally inclined and a systems biologist, so that's going to be what I'm the most interested in. But I think we really can benefit from embracing the DeSci movement for decentralized science and actually moving towards reanalyzing existing data rather than starting from scratch and saying, oh, we got $1 million grant, let's sequence 1,000 fresh whole genomes. If there's already genomic data out there, you might not have to start over. You might not have to invent the next new shiny something or new discovery or something from scratch if you can build on top of what's already there from existing work. And I think that's something we need to see a lot more of, but I think we are starting to see more of, especially with groups like Remission Biome rising and not to mention health rising. I sort of have this vision of seeing Court Johnson's blog Health Rising infused with horizontal data analysis, where you take existing resources and parse them and actually go from sort of a literature review type approach where you're using knowledge and conceptualization to then actually dig into data and evidence and say, hey, look, there's actually a database here that supports the claim by this study or this conversation. And I think that's something that it has a low barrier to entry that anyone can do. So I'm happy to follow up with people who might be interested in embracing systems biology and actually using resources that already exist to make progress.
Yeah, thank you, Sam. That's an excellent response. It highlights some of the very many challenges that occur in the field of genetics, and of course, there's all sorts of variables that impact the results. Different populations have different genetics as well. So, but yeah, making good use of genetic databases we already have would be really good utilization of funds, and hopefully something we do smarter in the future rather than working harder. Sometimes the smarter is the better approach. But yeah, thank you for sharing your experience and your story, and we'll get back to you. I'm sorry to keep you waiting, Dr. Jordan Vaughn. Are you there as well?
I am. If you can hear me?
Yeah, I can hear you. Thank you so much for joining me. Again, sorry about that wait. But I would like to introduce Dr. Vaughn as he's the founder and president of the Microvascular Research Foundation, which is a pretty awesome, incredible accomplishment. He's a physician and clinical researcher, and he's got a focus on clotting and vascular damage. And that's obviously a very popular area right now following, there's a recent publication showing that fibrin binds COVID spike protein, forming microclots. So a lot of patients are interested in that. If you wouldn't mind giving a little intro into your background and experience for everybody.
Yeah, so I'm just a classic kind of internist. I have, previously my undergrad's in chemical engineering, so I do have kind of a systems background to a lot of this as well. Kind of approach medicine a little differently because of that. I think one of the downsides of medical school right now is very little curiosity or questioning the current paradigms is actually, rewarded at all, much less in a academic sense or even to a research dollar sense. Instead, it's a lot of just falling in line with what the previous paradigms have been and just going along with them. So, what I really did was really treat people with COVID with a lot of things that actually Jacques Lobsher and Risha Pretorius had done in South Africa. And when I did that in acute COVID, targeting things like abnormal fibrin that had formed from fibrinogen because of the spike protein, I was able to salvage a lot of these patients and keep them out of the hospital, keep them from having to be on ventilators. Actually, pulled a bunch of people out of the hospital because, the worst thing that could happen was a lot of the ongoing treatments that were available there. And that kind of led me to having to figure out a lot of these people that did survive hospitalization that had ongoing or lingering issues, like how do we address those treatments? So A lot of what I did was basically utilizing the research that Risha Pretorius and Jocko had done, getting my own microscope, looking at blood, intervening with a lot of the recommendations they did, and then really kind of utilizing that information to tailor even my own treatments. So that is really kind of it's almost like a kind of a self trying to figure out how to help people right in front of me, and that's really what's led me down this pathway. And from that pathway, in addition, uh, the other things that have been seen are ME/CFS / long COVID. They all seem to fall in the same basket. But in addition, is learning about mast cells, learning about venous disease, learning about locations of mast cells, how they can be affected inappropriately, learning more and more about not only coagulation issues, but also fibrinolytic issues, the ability to get rid of clotting mechanisms. And so it's kind of opened up a whole new world that is quite interesting.
Yeah, on this vascular side of things, there's certainly a lot of publications out there on endothelial dysfunction in long COVID specifically, and I'm just wondering if you have any like uh, to say that these patients are having some type of vascular damage. I'm kind of wondering how can we characterize that damage? Like what type of damage? Because for people with EDS, for example, they might have laxity of like where their blood vessels stretch a lot, or like what type of damage are we seeing? Is there like a lot of blood vessel leakage, or like how can we characterize it?
Yeah, so a couple things. One is, in the permeability of the vascular system is also important. So endothelial damage causes abnormal permeability. In addition, a lot of these patients, especially patients that I see that have, a lot of them have compression syndromes or pelvic venous disease. They are typically people that have a history of, I don't know that you would typically define them as EDS, but they are definitely more flexible or hypermobile, a lot of them very athletic. And the more that we kind of dive down and look at the veins, the venous system seems to be, in the vascular system, the most immunogenic part of the system. And so the venules and veins actually, the veins, especially the big veins, even more, are covered in mast cells. And so when you get a lot of damage to the endothelium, you get a lot of shear stress on the veins, you get activation of mast cells inappropriately that cause a lot of additional dysfunction that can be multisystemic. And so that's the interesting thing that I've found is not to necessarily say that everything is caused by one root cause, but I will say most of these patients, instead of having 16 to 17 or even 30 diagnoses, are much more likely to have 15 to 30 symptoms caused by one or two to three things. And I think that is the cool thing to kind of put at the forefront and find out what that is. Now, not everybody has those issues. So that's the other thing within this huge basket. The basket is definitely separated by what I would call genetic predispositions, and figuring those genetic predispositions out, as Sam was talking earlier, I think is helpful for diagnosis and treatment. And I think one of the problems with research is going to be, because you have such a huge swath of people that are probably suffering from similar symptoms with very, not necessarily very different, but pretty different underlying causes. The especially the mono intervention or is going to be very hard to prove. It's also going to be very, it's going to be hard to tease out in the data as well.
Yeah, well, I'm glad you brought up the point of multiple causes, because as Sam had brought up, both he and I have a non-infectious onset of ME/CFS. So if I'm wondering, because in your opinion, if we assume that the spike protein in COVID causing microclots is a big component of vascular problems in long COVID. Do you have any ideas or explanations of how people without an infectious onset can develop microclots? Because microclots are seen in ME/CFS patients as
Yeah.
well. They might have different size or shape than long COVID patients, but is there that underlying mechanism? Obviously, that there are people who've gotten ME/CFS before COVID existed. So, what do you think the mechanism of that might be?
So, I mean, I think the term, as I think Sam already said, thromboinflammatory is what you have to look at. And the more that, or I guess the depth that we get to understanding how inflammatory cascades and inflammatory factors actually activate the thrombotic system, we can't look at them separately, if that makes sense. So even mast cells, I mean, actually, heparin, which we use a lot in medicine, was actually discovered because mast cells secrete it. So again, there's huge overlap in these systems, and I think one of the problems with our current system as well as our NIH is everything's so siloed that it really is not really how the body works. The body interacts on multiple levels with multiple things. We're not a siloed, we're not completely siloed like we like to think. Instead, pretty much everything affects everything else, and so that's how I look at it. A lot of these, whether it's a mold activated immune response, you're going to have a thrombotic response, especially if you're predisposed to having a thrombotic response. Does that make sense?
Yeah, absolutely. ME/CFS is a very complicated condition because it's not just like one particular system that's affected. I mean, we're seeing evidence of, the vascular system affected. We're seeing evidence of the nervous system affected. We're seeing evidence of the immune system affected. We're seeing the gut biome affected. We're seeing, everything you can think of. The more studies are done, the more things that we're seeing are affected. And I think the difficult thing is piecing together what's a downstream consequence of the underlying problem versus what's the underlying problem. So it's a good idea to get input from all these different backgrounds, like I'm trying to doing today to make sense of it all. But it is a very complex issue for sure. Okay, so I have some questions from by the way, thank you, Dr. Vaughn. I have some questions here from the general community. If you don't mind, I'd like to go through and ask a few of these. Just forewarning, I'm not able to, because I did get a lot of questions. We don't have time to go through every single one. And additionally, I'm kind of limiting it to questions that are related to science and research, specifically not getting into any advocacy or legislative stuff today, and things that our panelists might be able to speak to. So I apologize, can't get to everything, but of course, if you give our panelists a follow, you're welcome to tag them in tweets or something later if they're able to answer specifically. But yeah, so to jump right in, anybody can answer. I have a few notes here myself that I can talk about things a little bit, but I'm hoping somebody can speak up and we have a little bit of conversation going. So the first question I have here is on metformin. So the question is, some doctors are prescribing metformin, and I'm wondering if you've seen this worsen post-exertional malaise since it raises lactate levels. So metformin's a glucose inhibition medication typically for type 2 diabetes, but it also inhibits T cell proliferation, which reduces oxidative stress. And it's gained a lot of attention because there's been a study that showed that it decreased the risk of developing long COVID by about 40%. So there are people who want to take it preventatively. So especially, I guess, MDs in particular, if you have any opinions on prescribing metformin, people would love to hear.
Yeah, I do. So there's a couple of different things. One of the other things that I use a lot in long COVID is SGLT2 inhibitors. And I think what this points to is that there is a metabolic as well as insulin sensitivity dysfunction that's going on, especially at the microvascular area and even to the point that a lot of these patients benefit from those medicines, especially in the world of post-exertional malaise. Now, I will say I've had better success with SGLT2 inhibitors than metformin, which is a biguanide, but metformin actually, just kind of prefacing this, it's interesting. As I've gone through medicine as a chemical engineer, one thing I've realized is all of the medicines that we have are molecules and they act on cells. And just because you tell it or have been told that it acts this way doesn't limit its ability to act in every other way possible. In fact, that's what we usually call side effects. But in many ways, sometimes side effects can be beneficial when we're talking about other disease processes. One thing that I've seen in SGLT2 inhibitors, not only with helping with post-exertional malaise, is they seem to be a huge help to my patients with mast cell activation syndrome. And what I recently found was a paper in June that showed that mast cell activation and SGLT2 inhibitors, actually, there's a benefit there from down-regulating mast cell activation. So again, a lot of the things, again, as a doctor, we assume that what we're doing is intervening on the thing that we think we're intervening on. And I don't want everybody to question medicine here. But the funny thing is, a lot of times you find out the actual benefit to the medicine is even different than you thought it was. So even like metformin acts on platelets. It also acts on, natural killer cells, all of the above. So sometimes it takes the kind of physician to say, hey, this is weirdly helping a patient that I didn't expect it in an area that I didn't expect it to, and then actually kind of reverse engineering why that happened. And I think that is one of the things that's really useful in ME/CFS research, because instead of top down where we say, hey, this medicine's going to help everybody, instead you find this subset, this medicine seems to help a lot of people, and it's a little different than you would expect. And then you start to back up and say, what was unique about this subset that it did help?
If I could jump in there, Scott.
Yeah, absolutely. Thank you, Jordan.
And for any listeners, just to remind you that I'm not a medical doctor, do not give medical advice, and do not recommend any treatments for these ailments. But I was reading
all that to say that I think there's a component of metabolic dysfunction that is helped, which limits the damage to different areas of the body because you're on a biguanide like metformin. Just to throw that out there at the end as I got way off on a different topic.
No, yeah, I would absolutely agree with you on that point. I think you're absolutely right there. Sure.
I'd just like to point you to a recent review looking at all treatments for ME/CFS by the experts in clotting from South Africa and the UK. So they work for a major jabbing company, but they've published a lot of useful information on this particular subject. So they've reviewed all known treatments of this syndrome and its relation to COVID. Their names are Douglas Kell, Kell, and Tarasia, known as Rasia pretorius. And they were the ones who demonstrated that what they're calling fibrinolyds, the key focus of their research. And these are induced by, sorry to say, endotoxin. They showed that one molecule of endotoxin, lipid A, can trigger the polymerization of 100 million fibrin molecules to form alpha-beta deformed or amyloid clots. And so they're relating the blood chemistry and the treatments together with what they know. And so I'll add that review to one of my Substacks later today, but I just thought I'd throw that in, that there are two people here on X who you might like to follow. You can find them, Douglas Kell and Rasia pretorius, who've reviewed all treatments known for ME/CFS.
Yeah, thank you. I appreciate that. There's certainly a lot to be understood about, what triggers and what's the root cause, and how do we prevent these problems from occurring later on. By the way, I just wanted to ask Michael, did you have any input in this area? I saw you unmuted for a second. I just wanted to give you a chance if you wanted to talk.
No, I'm okay. Thank you.
Okay. All right. Sure. I'll go on to next question here. So one person asked, what future research is planned for analyzing connective tissue changes in ME/CFS and long COVID patients? And this is really interesting because this has obviously been an area of focus for a long time. Remission Biome just held they held a webinar with Jeff Wood. You can check out mechanicalbasis.org, where they talked about the subset of ME/CFS patients that have CCI. And obviously, a lot more work has to be done in terms of understanding what the root cause of these connective tissue alterations are. Because if they're not genetic, if it's, like we consider this to be an acquired problem, what is driving these changes? And so he's proposed activation of collagenases is to be one factor that can affect connective tissues, but it's something that's not proven yet. And so I think a lot still has to be understood and at least talked about and discussed, like what could be driving acquired connective tissue changes. So I'm wondering if anybody else here has any input on this.
So in a very small area that I'm looking into is venous compression disorders of the pelvis. So meaning things like iliac vein compression, May Thurner, other things like that. And it's interesting, a lot of these people develop it after exposure to the spike protein. And, most, again, the person who's my plumber, who does a lot of my endovascular treatments for this, argues or used to argue that, Jordan, they're born with this. And I don't disagree that they're born with laxity of the vessel, and I think it actually probably has some compressibility. But it's not fixed at the time. And one of the arguments I have is there's something happening to the endothelium during these infections, or during these exposures to whatever it is. In COVID, it's spike protein. And imagine that a vessel or an endothelium is already kissing, in a sense, the sides are touching. And when that endothelium gets activated, it's more likely to web and spur. And when we look at cadaver studies of people with May Thurner, the vessel kissing isn't, it has webbings in a lot of these people. It has spurs that basically cause the vessel to basically get stuck together. So that's, but again, that's not necessarily, something's fine. It can open and close fine, and then it can't. And it seems like there is an incident that causes it not to be able to do that. So that's just an example of some kind of dynamic, something that's dynamic, like whether a compression in the pelvis can open or close, can be impacted by an inflammatory response to some nidus.
Great, thank you. If anybody else wants to comment, just feel free to speak up. If not, I'll just move on to another question.
I'll add one aspect that's parallel to what was shared across a couple sections of this today. I guess it's sort of a hypothesis that that's been brewing in my head, following some of the research across long COVID, ME/CFS, and Ehlers-Danlos syndrome, EDS, is that there are these proteases that might be able to mechanistically connect the dots across mast cell activation disorders, mast cells and collagen directly, and collagen mutations in Ehlers-Danlos syndrome subtypes. And some of the specific research that's highlighted that includes the Norris Lab kallikrein gene mutations proposed for HEDS in a preprint released a few months ago, as well as just this past month or maybe two, another paper that this one's peer-reviewed, finding fibronectin fragments in HEDS specific that has high specificity to the hypermobile subtype of EDS. So sort of some innate immune involvement and some involvement and some interaction between the proteases that physiologically in a healthy setting would break down some connective tissue might be out of balance. So there might be some dysregulation or imbalance there that could mechanistically maybe be able to explain, once we develop further research, it's still a little early stage now, just how deep those connections are between sort of inducible connective tissue symptoms, connective tissue disorder symptoms. Inducible sort of being the first word that can come to mind, but the lived experience for many people with EDS, it shows a lot of intermittent stuff that you wouldn't expect to be so intermittent. And maybe mechanistically we can explain some of these sudden onsets and late onsets with some of these mechanistic interactions around proteases. Kallikrein being one of them, and the fibronectin fragments being something that I've seen pop up in a lot of different types of research now.
Well, yeah, that's you know, one thing I've thought about a lot is, there are a few researchers that have different theories on ME/CFS. Like Ron Davis has this metabolic trap theory that he's working on as an example. And, there's a few others. But to like my thoughts on it is, just because there's more than one theory for ME/CFS, there might be more than one theory that's actually true, and what they're proposing is actually going on, but there's also other things going on. So connecting that is really challenging. But with your regarding proteases affecting connective tissue and things like that, that should hopefully be something that I think you'd be able to spot in proteomic studies, which I know are happening. So if that's part of it, I hope that they'll be able to speak to that there. Okay, so moving on to the next question. Person in the community here has asked, what is your opinion on trialing existing rheumatological drugs such as interleukin and JAK inhibitors? And I know JAK inhibitors have got a little bit more attention recently. There's some ME/CFS researchers talking about it. I know Vincent Lombardi is studying JAK inhibitors, I think in ME/CFS currently. But there's also some concerns about them that they can increase risk of heart attack and stroke, and if it actually makes sense to use these in all patients. I think Ron Davis has previously shown that cytokine levels are elevated in mild ME/CFS patients, but actually depressed in severe patients. So might it be something that's better to use for one particular subset? I'm wondering if anybody else has any thoughts on rheumatological drugs.
I'm happy to opine.
Yeah, sure. Go ahead.
So, we had seen the use of tosatinib at Temptra when we had these very severe COVID patients that were initially hospitalized, and seeing responses to that. That being one of the JAK kinase inhibitors. The others we had used for a bit of time: baricitinib, ruxolitinib. I think as long as I think the way that we're going to go, one of the paradigms that I think we're going to have to go down with respect to attacking these disease processes such as ME/CFS, as long COVID, is that I'm a firm believer in that there's a strong immunomodulatory component to it. And I think the only real, one of the modalities, like we said, there's multiple pathways, multiple pathophysiological aspects that I think, treatments need to hit, because at this point, we're not 100% sure. That going down the route of immune suppression, to me, sounds very reasonable, as long as with anything in medicine, in clinical medicine in particular, the benefits outweigh the potential risks, and the patient is monitored very closely. I see it very reasonable that these, quote unquote, immunosuppressive agents, rituximab, Uplizna, even cyclophosphamide. There's some data to show efficacy in immune dysregulation in a cohort of ME/CFS patients that were receiving it for cancers, breast cancers and bladder cancers, I believe. So I think it's very reasonable, and I think, in fact, there are some trials that are going on right now, looking at baricitinib in combination with H1 and H2 blockers. Being that especially H2 blockers such as famotidine, beyond its antihistaminergic effects, does exert theoretically immunomodulatory effects, coupled with a very common antibiotic, minocycline. Another one where, beyond its antimicrobial properties, it's thought to exert a lot of anti-inflammatory and immunomodulatory properties. Particularly with that drug, it crosses the blood-brain barrier. And again, being that this is, in my hypothesis is very likely a central nervous system, amongst other things, process. You can see the efficacy of these agents, as well as others. For example, sirolimus, rapamune is another one that's being looked at. It's being administered in kind of an ascending pulse fashion of 1 milligram a week, then moving up to 2, ultimately going up to 6. And now that's looking at a different mechanism of immune suppression. That's inhibition of the mTOR, the mammalian target. But again, in terms of looking at these various modalities of immune suppression, I've personally, and I'm going, clinically speaking, personally witnessed, improvements. Obviously, depending on the degree of severity, the how long the patient's been sick, the age, the comorbidities, and so on and so forth. So I definitely think that's something that should be, of utmost importance to look at. That being, essentially immunomodulating medications, which yes, they are going to be immune suppressive, but again, if the benefits outweigh the risks, I'm fine with that. As a clinician and as an infectious disease person, I can closely survey these people for development of any infections. Thank you.
Yes, thank you. Any other comments from the other panelists? Okay, moving on. Another person here has asked about how prevalent is mold toxicity in chronic fatigue syndrome patients, and how is there any research that's going on in this area? And I've looked into this myself because first of all, I've done a survey in the MECFS community, and I just asked broadly how many people have problems with chemical or environmental sensitivities, and 80% of people self-identified as having them. So it's an extremely common thing. And I did come across one study that showed that mycotoxins were found in 92% of MECFS patients that are reporting chronic mold exposure. And this can actually lead into, I think, MECFS because these mycotoxins are known to lower T cell reserves, and this can lead to immunological complications that lead to we talked about problems with fibrin and vascular issues and all downstream effects. So I'm wondering if anybody else here has given thought to the role of mold toxicity and mycotoxins, and or have seen other people talk about it clinically, and to get some professional opinions on what patients are really talking about anecdotally. But I don't see much attention on the research front. So what do you guys think?
That's something that many of my patients have come to me with positive mycotoxin studies on whether it's Vibrant Wellness or any of the other tests, and what they think are clinical implications with that. And while again it is very likely patient dependent, and you need to see how many different individual factors are going on, I have no reservations with respect to implementing treatment, whether it be with a binding agent, a compounded intranasal biofilm disrupting agent, a compounded intranasal antifungal agent, and so on and so forth. So I have seen it in clinical practice, and I do believe it. I have seen some people improve upon implementation, although there are other factors in play as well. But I definitely do see it as a clinically relevant aspect.
Yes, Scott and Michael, have you used skin prick testing very much in your studies?
I have not done skin prick testing, as that's more to the allergists, in my opinion.
Yeah, so on the issue of the mycotoxins that Scott was talking about, I'd recommend to new listeners the United States Government Comparative Toxicogenomics database, which is based in North Carolina. It's a tremendous resource rather underutilized from what I can see. And you can search it on genes, obviously, but you can, for a specific chemical or a specific allergen or a specific disease, and you can find a wealth of information there, more than any one person can cope with. So if you haven't been there before, have a look for the CTD, as it's abbreviated.
Great, thank you. If you have any links, if you could post it in the chat, there's a bunch of people following all the comments. So I've been posting some of the links to things that I've been talking about in the comment section as well. All right. Hey, I had another question here specifically for Sam. Sam, are you still around?
Yes.
Yeah. So I just kind of had to ask this of you because AI has become such a huge thing that I hear just on the news constantly. Everybody talking about hugely being funded by Google and Facebook and Salesforce, and all these huge companies are putting billions and billions of dollars into these new AI systems. And that's actually been a point that I've heard has given a lot of patients hope that even though the progress of medical research has been really, really slow, having the potential to use a tool like AI to help speed up research. And so I'm just wondering, like, what your, if you have, because you have a computational background, what role you see AI playing in ME/CFS research. I'm sure a lot of people listening would like to hear. And if you've seen any researchers that have begun to implement that technology yet.
Well, for starters, I'm certainly trying to. I have huge ambitions for deploying AI and LLM era tools, LLMs being large language models like ChatGPT, far beyond the infrastructure of chatbots into just sort of these workhorses, where I think we've got plenty of potential and hope to see AI actually catalyze progress. And one of the things that sort of strings together what a bunch of us on this panel have talked about today is sort of how we frame knowledge, how we comprehend labels on an individual level and communicate them. And ultimately, what I have hope for on the LLM front is this sort of vision of knowledge aggregation, where we can take everything that is accessible to pour in as training data or input data, and actually be able to parse in a much higher throughput than any human effort can ever reach. Since that much is proven that AI can be much higher throughput if you can get the right code and the right script and the right structure engineered. And basically be able to do some knowledge aggregation, like I alluded to earlier, around biomarkers and subtypes, but also just claims and evidence. If you can map together, hey, it looks like there's this knowledge from a completely separate label, whether it's a separate condition or a slightly different protein that's under the same pathway. If you can actually identify that algorithmically, computationally, then you've got a highway to high throughput progress. So I actually see room for not only decentralizing and crowdsourcing science and research and progress, but also being able to accelerate and catalyze progress with AI. And I think there's a beautiful room and space for the synergy between the two that I'm hoping to launch ideally in the near future. Have to finish designing and structuring it, but sort of room for systems biology, AI, meeting in the middle to actually catalyze progress with existing resources so you don't have to start from scratch, just in novel combinations. And I guess one thing I can sort of provide as context from my 2 years of industry experience as a computational biologist, I was sort of tasked to be a glorified software installer. But what was really neat about that is even after years of training on how to code, I rarely had to write my own programming code at all. I just built pipelines that deployed existing solutions into novel combinations. And I think that's something that I have sort of started to see beyond just the one job I'd spent that time doing in that framework, where we can take existing solution frameworks, existing problem spaces, and make novel combinations where it's not necessarily discovering a new problem space or description of it, or even understanding of it. You don't have to necessarily make a new discovery or invention from the ground. You just have to make a novel combination of building blocks. And I think that's something that AI gives us huge potential to do.
Well, in terms of getting immediate accessibility and affordability for, these AI systems, which can be very costly to develop and even more costly to run them, they consume a lot of energy. You think there's potential for, in the near term future, some of these smaller research labs to be able to partner with some of these large tech companies like Google, to be able to utilize their AI systems on specific projects that we can get, an advancement in our research production, sooner rather than later in a cost affordable way.
Yeah, I think that's something that would, in the end, come down to an advocacy mission, that you have to be able to scientifically communicate your mission statement and uh, problem state, basically how high the stack of unmet needs truly is something I tend to refer to a lot across the board, especially looking horizontally. Because if you're speaking different languages about different problems, if you can compare the unmet needs and maybe find common ground, you might be able to get sponsorship. You might be able to get common mission alignment across different orgs. So I can't say that I've got a ton of experience with getting the Googles and the giant companies of the world behind me, but I certainly have hope that AI is giving us a catalyst for progress there.
Great, thank you. Yeah, I appreciate those comments. I know it's a very well-funded area field now, and I hope can be a big help in research. I also had a specific question here for Dr. Scoma, if you don't mind hopping back in for a second.
Sure.
Yeah, I just have to ask you because there's so many people that talk about this on X, on Twitter, that there has to be time dedicated to this subject today. Is the role of viral persistence in these illnesses? Because specifically, you have a background in infectious disease, so I'm hoping you can speak to this because I've seen studies that have shown in long COVID, right, 43% of patients show viral antigen persistence, and then in the ME/CFS population, virtually all patients have uh, EBV reactivation or HHV sex positive, something like that. And so people are trying to figure out to what degree those the viral persistence is meaningful, if it's actually causing symptoms or if it's just like a sign of under functioning immune system. And additionally, I just kind of wanted to ask you a second question that kind of pairs with this.
Sure.
A lot of people have come to me and said that antihistamines and mast cell stabilizers have helped them a lot. Obviously, there's many people that they do not help. So I'm just wondering clinically, like what percent of your patients respond favorably to antihistamines, and then additionally, like antivirals, like what percentage of these chronically ill patients are antivirals and antibiotics beneficial for, because it's not every single patient, not everybody responds positively to it, but how much is it worthwhile for people to try things like that?
Okay, so sure. So thank you, Scott. So I'll answer each one in a row. Obviously, given the ability for coronaviruses to infect and reinfect, individuals over a time, as you said, it certainly seems to play a role, but I don't think that that's the, sole aspect at play. As you said, there is ample evidence in some studies that shows persistence of viral RNA and proteins that have been isolated from specific tissues, such as the respiratory tract, the GI tract, the central nervous system, presence of antigen. Now, does that, presumably that's going to portend to that degree of incomplete immunity and holes in really the immune response, namely, B cells, T cells, natural killer cells, and so on and so forth. As that pertains to antivirals, I don't think we've seen a whole lot of success yet in antivirals, specifically such as Paxlovid or something of that sort, for a longer stretch in long COVID infections. I haven't used them in that setting outside of the acute illnesses. And again, some people just have a difficult time tolerating them. So it remains to be seen. I think that's something that still needs to be looked at and studied further. There are other antivirals as well that are just, shelved by the FDA and are not even being used. And, in some sort of a desperate situation with respect to people that are, truly, truly suffering from these conditions such as long COVID or ME/CFS. I think all things need to be on the table with respect to treatments, as long as, again, they can be looked at and implemented in a safe manner. With respect to antivirals in ME/CFS, this is something that has been looked at for quite some time, coming out of Stanford and so on and so forth. Everything has its place. Roughly, I've seen it help roughly about 50% of my patient populace. Again, if you're talking about specific antivirals, I have not seen acyclovir or valacyclovir to be of much help. Unless they have issues of, say, maybe VZV reactivation, as a couple of my patients do, because they simply do not cover CMV or EBV or HHV6, which are, it seemed to me to be some of the bigger players. So again, I'd put it at around 50-50. It's certainly, a patient needs to be on a reasonable dose, in which I don't want to get into doses on here, but happy to opine otherwise, as well as they need to be on a reasonable duration of time with surveillance, with an intermittent CBC, Chem19. Mind you, these are medications that transplant recipients, of which I take care of all the time, and I trained in a transplant center, that being Yale. They're on some of these medications for the rest of their life. So these are reasonably safe medications. And then as to your last question with respect to mast cell is difficult because it's very difficult to diagnose. All of these inflammatory, all of these mediators that are, of which there's, hundreds to thousands that are released by mast cells, they degrade so quickly that even you can have somebody who comes in with symptomatology pretty consistent with mast cell, and I'm not just talking about cutaneous reactions or new onset food intolerances. I'm talking neurological issues, migraines when they never had migraines before, brain fog, new onset anxiety and panic attacks, GI issues, pulmonary issues, joint aches, muscle aches, you name it, small fiber neuropathy. But it can be pretty difficult to diagnose, even when somebody is in a quote unquote flare, for the simple fact that things degrade so quickly. So I usually do tend to at least have a H1, H2 backbone in the majority of regimens. And I've seen people improve. Problem is that usually I'm constructing regimens that are, I try to minimize polypharmacy as much as possible, but, people that are under, a number of medications, although at the same time, I do like to kind of implement things in a stepwise fashion, such that if people are going to have an adverse effect or a positive effect, we have a better sense of things. And that portends to that trial and error aspect, which is just an unfortunate aspect to this disease, because it's different than, say, the patient who comes into the hospital with pneumococcal pneumonia and septic shock. Yes, they may be on death's door at one point, but at least the process that they're incurring is more straightforward. You know how to treat it, and they get better. These, chronic fatiguing, chronic neuroinflammatory, microvascular immunologic conditions to me represent the greatest challenge I think that there is, which is why it drives me so much, in particular with working with these patients. So again, not to get off topic or to beleaguer the question, but I've seen efficacy with implementation of mast cell, in particular sometimes when people have severe POTS and or dysautonomia type symptoms, and you trial them on an alpha blocker and a beta blocker and clonidine and droxidopa and everything else, and they don't tolerate it, and then you just kind of ease them into an H1 H2 blockade regimen with maybe some cromolyn, all of a sudden their dysautonomia just goes away. So it's probably mast cell related, of which, as our additional panelists spoke to, mast cells line every aspect of the body. So it's very, very all encompassing.
Yeah, I think a lot of people listening find that information really, really useful. Uh, I wish something like this talk has happened in previous years, that people could learn from this, and the doctors that find, I guess I'll say anecdotal evidence, because you're not performing a clinical trial when you present this information, but it's really useful because you handle hundreds of patients over the years, and it's really, really helpful to be able to get that. And I think that's why a lot of people end up on platforms like X and Facebook and social media, and using that as like a primary tool for their medical care, because getting input from other patients online is like really the only place they're getting them. Because if you go to a specialist that has no experience in ME/CFS, they really have no help. They have no knowledge, and so patients are kind of forced to get it on their own. So information like that is super, super helpful.
I've never seen more of a well-informed, well-researched patient populace than this patient populace, as you just said, because of the simple fact that they don't have resources elsewhere. The majority of people who I see have been through a dozen providers. And at least I think if a provider is at least open to learning, open to researching, open to patient suggestions, that's fine. But the degree of just ignorance and gaslighting and so on and so forth that goes on, it's just repugnant, especially when you're seeing this subset of this group of patients that are just incurring such a degree of just suffering. I've never seen anything really like it. You're talking about somebody, in the prime of their life, graduating college, magna cum laude, for example, and they go and get a virus. They have 105 fever. They just never get better after 3 or 4 years, and they're 26 years old, bedbound, not even ambulatory, and they're some of the most educated people you'll ever want to speak to. I learned from these patients, just like they learned from me, because really, if you're bedbound and compromised to that degree, what else are you going to do other than go onto these forums and research and be an advocate for yourself? So if I can be, for example, without being too getting too philosophical here, if I could be an advocate for this patient populace as a provider, that's a good quarter of the job done right there.
Excellent. Yes, thank you very much. We definitely appreciate your support. I just wanted to make a note that Dr. Vaughn had to step out. He had some family matter to deal with. I certainly understand, but I appreciate him giving his time, and I thank him very much. I won't take too much longer, and I'll wrap up in a little bit. I had one more question, Geoff, if you're still there, that I would like to ask you specifically. And then I just have a general wrap-up subject for the discussion, and then we'll end up after that, so we won't take too much longer. But hi, Geoff. I have a question for you. I saw posted about Zeolite the other day. You made a post on X about Zeolite, and I was interested in that because that was something I was recommended to a couple of years ago that I tried for myself as like a supplement, and because it was advertised, and other people on the internet had said, oh, I'd use it, it's harmless, whatever. And so I tried that, and it alleviated some symptoms for me initially, but then caused other things to get worse, and I discontinued that. And so I'm hoping that can you speak to the hazards of, I don't want to say just zeolite, but kind of using some of these unapproved treatments, off the cuff, just as a word of caution to the patient population, because I understand when you're very sick, you're very, very desperate, but certainly we don't want people to take a bad situation and make it worse. So you posted about that, so I'm wondering if you could speak to that.
Yes, thanks, Scott. As I said, I don't recommend treatments, but I point to the extreme danger of lunatic treatments like the zeolites. One in particular is being flogged by profiteers to squirt into the mouth of not only yourself but your pets, and other people are selling packets of zeolite to swallow in relatively large quantities. So I was a founding member of the Mineralogical Society of Victoria here in Australia, and I visited the back rooms of the mineralogical division of our museums, and we studied in great detail. I became very interested in the crystal structures of zeolites in particular, and they're extremely dangerous, fibrous, sharp crystals, and they often come as a mixture of minerals. The so-called purity of the stuff being flogged is highly questionable. They basically behave very similarly to asbestos. So if you want to give yourself asbestos, mesothelioma, or colorectal cancer, then you would consume zeolites. Otherwise, you should stay away from them. You can appreciate the beauty of their crystalline forms and their internal structures. They have industrial uses as catalysts for making gasoline and various other processes, but deliberately exposing yourself to aluminosilicates is madness, in my opinion.
Yeah, thank you. I also saw when I had made a post about this space. One of the things that I saw we're hoping to talk about is sick building syndrome, and that's something, because of my chemical sensitivities, I definitely suffer from a lot. And so, yeah, I'm wondering if you had any things that you want to talk about regarding sick building syndrome, which is often synonymous with ME/CFS.
Yes, so the primary cause of sick building syndrome is, you'll be surprised to hear, endotoxins. So a great deal of research has been linked to that, and a lot of sick building syndrome came from cost cutting and reduced cleaning. So cleaning is essential to remove shed endotoxin wherever humans congregate. But endotoxin is also in buildings from cigarette smoke and marijuana smoke. That's one of the major causes of symptoms of ME/CFS. So that there's then a more complicated story of moving from short-term exposure to endotoxin in sick buildings to long-term. So there was a study done after the tsunamis in Japan, where large numbers of people had to be put into emergency housing together, and the resulting mass sickness that occurred was directly related to the sudden increase in exposure to other people's endotoxins, simply because of the high density in the emergency shelters. So I've put up some links to some of that sort of story. So it's a very interesting thing in the relating directly to the ME/CFS. I posted today a sickness questionnaire mean scores graph that I found, where they looked at the psychology of the sufferers and compared it with people who were deliberately injected with endotoxin 1.5 hours afterwards, and they then measured the psychological factors. But there's been a huge amount of work done on sickness syndrome or sickness disease it's got various names from both short-term and long-term exposures. So it's I've got a Substack on that sickness syndrome. A lot of that research is done in Australia in combination with scientists in Sweden, and definitely worth following up if you're interested in sick buildings. So I'll do a more detailed Substack on that eventually, but there's a very large literature that needs to be sifted through and sorted. So, but it's certainly cause and effect is well documented.
Yeah, if you make a post on sick building syndrome in particular, I'd love to check that out. So yeah, I appreciate your time and explanations there. And I'm just going to wrap up in a couple of minutes here, but I kind of wanted to take the end just to go with the panelists that I have here one at a time. Somebody asked from the community, and obviously this is like probably the number one thing that I get asked in general. What do you consider the most promising treatment for ME/CFS to be? And I think whatever the most promising treatment right now is going to be different than what we think the most upcoming promising treatment would be. But I think it's also a good opportunity to, if anybody wants to make like closing comments on not just like what we think is promising, but where we think ME/CFS research is falling short, and what we can learn from, like I recently listened to the NIH held the TLC, the long COVID recover event, and that was really, really huge. And all the long COVID researchers there, they across this 3-day period, they hardly brought up the subject of ME/CFS. And I think that's a shame, because ME/CFS has been around for decades, and there is a lot to learn from this. So yeah, in addition to what treatment you think is most promising, where do you think we're falling short, and where can we learn from?
If I could start, and I want to thank you for having me and the panelists, and just want to apologize for the initial technical glitch. I think with respect to where we're heading with respect to some of the treatments, I think with respect to immunomodulating therapies, like what I had expounded on earlier, I think are going to be should theoretically play a huge role, being that this is very likely a derangement of the immune system, and a neuroinflammatory process, which to that point, I think there are other things that can be looked at. They have to a small degree, but need to be looked at better with respect to even some of the treatments for some of the neurological syndromes, such as MS and ALS and myasthenia. And there are small little cohorts of patients that have been looked at with respect to agents such as efgartigimod or ibutylast, which is a phosphodiesterase inhibitor, Vercuov, dalfampridine, agents of that sort. So that would be an interesting spot to look at, as well as the use of some of the agents such as pentoxifylline, for example, which is a xanthine oxidase derivative, with respect to not just ME/CFS, but long COVID as well, both with respect to its anti-microclotting properties, as well as what they are now postulating to be some degree of even immunomodulatory and/or immune suppressive properties. So I think that there's certainly a lot to look at here, but it's obviously has to be backed by better studies and better data. Which again, it's amazing to just think that you have a disease that has such a high prevalence, 2 to 3 times the prevalence of that of processes such as rheumatoid arthritis and so on and so forth. Yet we don't have really any biomarker for it. I know that would probably be another whole topic of discussion. We don't have specific biomarkers for it. We don't have specific defined pathophysiological or genetic mechanisms for it. And I think only once we get that are we going to really be able to have targeted therapies. Right now, it's essentially repurposing of FDA-approved medications and/or supplements. Oxaloacetate is a very good one. I've seen a lot of improvement with that, although it's very, very prohibitively expensive. But we obviously need the enhanced research in order to continue to try to move forward. Because I definitely have seen, the more severe someone is, and the longer they've been sick, a little bit less, better the more deleterious of prognosis for them.
Great, thank you very much. I appreciate that. Geoff and Sam, do you have any comments?
Yes, I do. Just to follow up on what Michael was saying, there are in fact very promising markers for ME/CFS, and it's a pity that Jordan had to leave because he's one of the leaders in the field and is part of an extremely large research team. They've related ME/CFS to things like soluble CD14, WASF3, which was new to me, but certainly something I just posted about on my X space. IgM, immunoglobulin M, lipopolysaccharide binding protein is directly linked to ME/CFS as a marker. So is interleukin 8, which is a sign of endotoxin tolerance, which is an extremely complicated process that I was sort of touching on very briefly before. You have your short-term reaction to endotoxin, whether it's through gut dysbiosis or inhalation or jabbing, and then you can develop what's called endotoxin tolerance. And what happens there is that your immune system natural defenses are collapsed against new intrusions, and you get this a dysbiosis of your cytokine expression profiles. So if we can get Jordan back on another day to talk about his detailed studies and his large group studies of the markers, that'd be really useful.
That's good to know. Yeah, I'm sorry he had to go. Family issues come up, but hopefully he can at least give a comment in the comment section later on. Sam, did you have any final comments?
I think a lot just similar to what I've been emphasizing, just to circle in on the hope I have from the DeSci movement for decentralized science of basically between systems biology and decentralized science and just the sheer amount of motivation and passion and skill in the ME/CFS population. I think we have a great potential to empower the patients and the whole population of researchers, clinicians, patients, and beyond impacted by ME/CFS to actually make a difference by catalyzing progress and including and empowering people who have all that motivation, all of the skills that they've built up over years of struggling through and not having any resources or FDA-approved drugs. So I'm certainly a big fan and hopeful that we'll be able to speed up research and progress in treatment development. And I guess on the clinical side, even though I'm not a clinician myself, one research group I just wanted to point out is that of Norway. The same research group with Dr. Flug and Dr. Tronstad that published the rituximab clinical trial for ME/CFS has also put out a more recent, I guess I'm not sure if the paper from this July is exactly the same as what was presented last year out of the Berlin Charité ME/CFS conference. But I have a lot of hope in their focus on the inclusion criteria of severe ME. So actually taking cancer therapeutics like rituximab that depletes B cells with the high risk profile that carries and weighing that with the huge unmet needs for ME/CFS patients, including severe ME. And I think definitely keep an eye out on what they are putting out. I don't think I'm allowed to share everything that was presented last year in Berlin, but definitely keep an eye out for when they do publish that. And there are drugs that they are trialing with full RCTs that are looking more promising than even the early stage data did with rituximab.
Yeah, excellent points. Definitely inclusion is an important thing to consider, and severe ME/CFS patients haven't been included in a lot of studies. And as we've seen, like with Ron Davis, his cytokine study I mentioned earlier, their biomarkers can differ in totally the opposite direction of what mild patients have, and there's a lot of heterogeneity as well. I just wanted to also give my personal input as a final comment too, in regards to what I consider the most promising treatments to be. I've noticed in not just ME/CFS, but a lot of long COVID patients, air quality is really important, and I think that the importance of that relates to reducing toxin load and elimination of triggers, and that's something that can impact mast cells and things like that. And I found that not just in myself but in the community to be really important. I think in the future, some things like there could be a blood filtration therapy that we could develop, because there's so many things that we find that are harmful, whether it's viral persistence or endotoxins or blood clots, etc. To be able to remove those from the blood with a filtration system, that would be beneficial. I think like with myself, I think of chemical exposures, but in the long COVID community, COVID's a respiratory virus, and I've put a lot of focus on barrier damage. So the respiratory epithelium is actually destroyed by SARS-CoV-2. And there's a few papers on this. I'll post one in chat right now. And nebulizing stem cells would be a targeted therapy that could help repair that barrier damage specifically at that specific site. And so that's something that I think could be interesting. And also in Spiritol, there's a trial for like an inhalable antioxidant mix. I know that's kind of generic, but I've heard good things about it so far. And it's the only like ME/CFS trial that I'm actually following. So those are the kind of areas that I'm interested in terms of therapeutics. But in terms of where ME/CFS research has fallen short, I think that ME/CFS researchers have definitely not done the best job at listening to the community in the past, particularly with like anecdotal evidence, which I think is actually really, really critical. And then what I'm seeing in long COVID researchers is that a lot of non-infectious patients, not people with non-infectious onset to the illness, are being excluded from all discussions that's happening with long COVID. Because right now we have this infectious associated disease or post-infectious associated disease, and not to underscore, infections are obviously really important if you've had an infectious onset, and viruses do cause damage, absolutely. But I think there's also critical clues that people without infectious onset are still having this condition, and that there's an overlap between those two groups. And I actually think that's an important clue, and I hope that long COVID researchers do learn from ME/CFS research in the past and are inclusive of all the different patient populations, and that there's hopefully better communication in the future, and really considering these anecdotal reports, which I think are really, really, really important and have biological relevance. So, yeah, that's just my personal input. But I want to end it here. It's been full 2 hours, like on the dot. I'm impressed I was able to get that time so well. But I wanted to thank, if we can give a big, big, big thank you to all the panelists that took time out of their day to join us on this call and share their knowledge and stuff they're doing in their research and these articles. you all have been awesome. So if people in the chat could either throw up a heart emoji or just type a thank you in the comments, I know our panelists will really, really appreciate that. So I just want to say thank you to you guys very, very much. And anybody that's listening in, please give our panelists a follow. And I will also post a recording of this chat. So if you weren't able to listen to the whole thing, I'll have the recording posted so that you'll be able to go back and listen in on key areas that you were interested in. So thank you again very, very much. And I hope to have you guys either back on soon or hear more about different research and medical areas that you're studying, because you've been a tremendous help. So you guys have been really, really great. Thank you very much. All right. Bye, everybody. Have a good night. I'm going to end the space here.